I didn't know that I was bitten by a mosquito last night, but when I woke up and found a drop of blood on my aqua blue bedsheet, I knew. Am I going to get malaria? Am I going to die? Before I answer that, let's look at how one gets malaria. When you are bitten by a mosquito, if the mosquito that bites you has a Plasmodium parasite, you stand a chance of developing malaria, obviously. In this part of the world, Ghana, where I am, it is Plasmodium falciparum that is mainly responsible for spreading the disease. Within minutes to hours of the parasite entering your bloodstream, it quickly makes its way to your liver cells. There it hides and divides until it gets to a stage called a schizont. This is literally the stage where it's made lots of copies of itself. Then it bursts out and quickly infects red blood cells. It does all these things quickly because if it hangs around too long, your body's defense system, your immune system, will detect, attack, and destroy it. But I have a question. Why the liver? Is the liver immunologically privileged, as in it doesn't get attacked? That's a question for you, my dear viewer. So back to the infection. So what happens inside the red blood cell? And these concepts are credit to Kirk Deach, who gave this talk at the Johns Hopkins Malaria Research Institute in 2003. His video is online. Taking excerpts from his video in order to look deeply at the methodology, the techniques, because that's what we are interested in on this channel. So the parasite's inside the red blood cell. In the red blood cell, we can crudely, and some people will be offended, some experts will be offended, but we can crudely think of it as a bag of hemoglobin that these parasites are living in. That's the reason why they infect the red blood cells. The parasites, when they're there, import and export proteins all the time. They do it to make modifications to the red cell. The cell is very biconcave in its healthy natural form. It's very flexible and it can squeeze through all your capillaries. But when the parasites invade and start setting up shop by importing and exporting proteins, that red cell becomes rigid. It becomes spherical. And at this point, if that red cell goes through your spleen will be like, hey, what is going on here? And it will clear the parasite from the circulation, destroying it. So the parasite has developed ways to avoid going through your spleen. So look at this electron micrograph, which shows you, like some of these, scientists have viewed this. So you see these knobs that are on the surface, making it look rather jagged. So here's another electron micrograph where you see the parasite in the upper right hand corner. You can see the double membrane. It has a double membrane. One is for the plasma membrane of the red cell, and then the other is the parasitophorous vacuole membrane that the parasite envelopes itself in. And what this knob really allows it to do is to interact with the endothelial cell. And in this way, it's able to get a bit sticky and adhere to the blood vessel wall. And that's how it avoids going to your spleen. And so interestingly, when clinicians look at people with malaria, falciparum malaria, they really only see the young form or the front end of the cycle, the parasite cycle in the peripheral circulation, because all the mature stages are actually tucked away and you don't see them in a blood smear. I have a video on here on how blood smears are used to detect the presence of malaria or Plasmodium falciparum in people's blood, so you can take a look and you can actually see the different stages that the parasite goes through, which you wouldn't see in mature stages in the blood cell, in the blood that you take from a patient. What I show you in the video is actually cultured cells in the laboratory, and this is why having cell lines are important, because then you can actually see all the stages of the parasite and study it. So this avoidance of the spleen, as you can imagine, is great for the parasite, but it's really bad for you because it means you can have your blood vessels clogged up with these adhesive infected cells that bind to your capillary endothelium, causing a lot of inflammation and a lot of problems in your circulation. This is classic for pregnancy-associated malaria. And then the other example is cerebral malaria, cerebral meaning the brain, where the binding is in the brain and you get a lot of inflammation and brain swelling. These are severe cases. So one of the bad things about having malaria is this anemia that you get, and that's literally because the parasite is bursting your red blood cells. As it makes more copies of its cells, it bursts. So I have a video on here about the life cycle. So as it bursts, it finds new red cells, it goes in there, starts multiplying, starts growing, bursts again. So it's doing all this bursting of your red blood cells. So it's no wonder you get anemia. But the other pathogenesis or bad things that it does that we're looking at in this video is this tucking away in your blood vessels, which is called technically sequestration. So this sequestration has been studied academically. And so here's an article, a review article from Michael Good and Lou Miller, where they show the progression of disease in an infected person. And at day 0, they intentionally do not treat and let the infection go over a course of like 200 plus days. It's only after the 200 plus days that they give treatment. And so they're able to observe what's happening within the 0 to 200 plus days. And so what they see and what humans with malaria manifest is this rising and falling waves of parasitemia over time. At day 0 of the infection, it's progressing. You can see a peak of parasitemia. And this is parasitemia is referring to how much parasite is in your red blood cells. And in this case, they see 100,000 y'all, parasites per microliter of blood. To give some context though, in 1 microliter of blood, you typically have 4.2 to 5.9 million cells. So the fact that 100,000 cells are in per microliter of blood indicates to you the level of infection that you have. So in what's considered low or uncomplicated malaria, you typically have like anywhere from 10,000 to 100,000 parasites per microliter of blood. But where you're getting more, like 100,000 to 500,000 parasites per microliter, these are considered high or severe malaria. And then hyperparasitemia, this is when it's like off the chain. You have greater than 500,000 parasites per microliter of blood, and this is greater than 10% parasitemia. When the parasite infects a healthy human, the human will defend itself. So they will mount an immune response and an antibody response that drives the infection and those parasites down. And so the parasites begin to get cleared. But what you see is that the parasites change their antigenicity and then they rebound. So you get a second wave of parasitemia. Antibodies arrive again against the wave and you clear it. But once again, there's recurrent waves over time. And this is classic antigenic variation. So what's happening? What's allowing the parasite to exhibit this variation as you attack it? There are multiple proteins that go through the red blood cell on the surface and on the outside where antibody responses recognize it. The one we're looking at in this video is thought to be the dominant antigen that is used by Plasmodium falciparum to evade your immune system. And this protein is called Plasmodium falciparum erythrocyte membrane protein number 1. So let's break it down. So the name PFEMP1, so Plasmodium falciparum, that's the organism we're talking about. Erythrocyte, it's the red cell we're talking about, so that's in the name. And it's a membrane protein. And because I mentioned there are multiple proteins, it's the membrane protein number 1. And this has a single transmembrane domain that extends out and interacts with host endothelial receptors in order to help it stay tucked away in blood vessels. There's a lot more to cover about this protein and how the parasite uses it to avoid being cleared. But this video is getting long, so I'm going to end it here. Thank you for giving it a thumbs up if it's helpful to you, and look out for part 2.